Yeah, so my name is Gyorgyi Tuba. I am a research associate with Laurentian, and I have been forever. And contrary to what's in the agenda, I am not going to be the main presenter of this presentation. But since I'm up here, I just wanted to introduce this project a little bit and explain what it is about. So we are one of the groups who are working on a specific aspect for the synthesis part of the Metal Earth project, right? So it's a joint effort of quite a few of us, and there are GIS aspects and data science aspects and geochemical aspects how we gonna handle this project. So basically what we do is we take all the geochem data that Metal Earth has accumulated in the past 5-6 years, and ultimately we wanna come up with ideas to be able to compare the ABTB with the Wabigoon. So it's an endowment / fertility project.
Use the microphone.
I do.
No, you're not. Mine was complaining.
Is it good now? Oh, awesome. Okay, so, oh yeah, I can hear myself now. So basically, this presentation will be on a specific part of our project, and this is the how are we getting to the point where we can actually start the project. Because as you can imagine, this database is not really the grab and go kind. So even though it's within one research group, an extended research group, these data were accumulated by over 50 people, and it's quite diverse in many of the senses, and Ahmad's going to talk about that in a little bit more detail. So we had to process this database quite extensively before we arrived at this point where we can actually start looking at the data from a statistical and the geochemical point of view. But why we put together this presentation is that I think this is going to be very beneficial, because I'm sure that you either have worked or will be working with legacy data. So this database is pretty much a legacy data. You come across this in industry. You come across this as an academic geoscientist, where you have to compile historical data from different databases analyzed with different methods. And you will have the same kind of pain as we had with coming up with a more robust and concise database that we can start working with. So this is going to be very beneficial, and also we wanted you to feel our pain a little bit. And yeah, I'm gonna let Ahmad speak for the most part of this presentation, and I will be back a little bit with my part.
Hello, everyone. I'm Ahmad. As you see, I'm going to present this talk on Lithogeochemistry Compilation dataset collected over many years in Metal Earth project. And this is the update that has been under progress, and we are going to process the data. So Metal Earth geochemical data compilation is we are going to systematically collect, process, and integrate high-quality geochemical data, ensure that the quality, precision, and accuracy of data are okay, ensure necessary data imputation and prediction of missing values and below detection limits, and make the finalized data set available for every end user, industry researcher, whoever wants it. And the ultimate goal in our project is, the geochemical synthesis project is to compare and contrast the fertile ABTB versus less endowed Wabigoon subprovinces using geochemical data collected across different transects, well, mainly transects and also the thematic ones. This is the map showing the location of samples, the name of the transects, the name of the thematic projects, color-coded to show the differences. It's too small here to be seen, but it's available online. You can see the spatial position of the samples, and the samples collected over 5 years or more with different geologists, more than 50 geologists, spanning large areas. There's a bias on that in spatial and geographical distribution sampling campaign. Targeted a variety of geological problems from stratigraphy to mineralization, and it's gone under numerous analytical procedures analyzing variety of analytes and components. So it's not easy to use. If you want to use that one, it must be homogenized and harmonized. So after that, we can use it, or end users can use that one. The left bar chart shows the distribution of the samples collected over Metal Earth Transects and color-coded for different classes of samples, certified reference materials or reference materials, or different samples collected for each transect, and they were available in the analysis list returned from ALS. Well, we need to consolidate them. We have one sample analyzed with different schemes to have actually good accuracy and precision for a specific analyte, so they need to be consolidated. This is completed, and we have a matrix of data which can be used and ready to go. ALS lab field duplicates were removed from the QA. We collected all spreadsheets and extracted the proper sample analysis in one spreadsheet. And each transect spreadsheet includes multiple compilation sheets, metadata, methods, detection limits, and different information which are available and accessible. The final data set contains 4,483 samples with unique values for each element. And in this one, we see that we have 110 reports from ALS collected, and you can find the number of samples as a bar chart here representing each transect. This graph shows the Metal Earth thematic projects, same as Transect. We can see the number of samples color-coded by the type of sample analyzed over the thematic projects. Well, the first step in order to start analysis and processing the data is to make sure that the quality of the data are okay. So we employed Thomson and Haworth's plots in order to see the precision of the analysis. Well, this Thomson and Haworth charts accounts for heteroscedasticity data which are available in geochemical data set, and it turned that most of the data that we have, they have a good precision. We see these two lines here. The more data points below these two lines shows that we have better precision in data set. So for example, for these two analytes, we see the most of the red samples, which are lab duplicates, they are sitting below the lines, and they're suggesting that the precision is in good order and good shape. And for most of the elements, we have the same situation. But if we want to use any element in our processing, we have to go back for the quality control report and make sure that it has a proper precision and then use it. And it's been the case in pre-processing and processing the analysis that we have started. Well, the next step was check the accuracy of data. There were lots of reference materials. We employed 3 of these reference materials spanning different chemical composition of rocks. As you see, these are the reference materials. They were very helpful to pick up the errors. We had some mislabeling, switching the sample errors, and we picked up them, we fixed them, and if there were a problem and we couldn't solve it, we removed that sample from the data set. And apart from these few samples we see here, most of the samples that analyzed as a reference material are in the good shape and within the boundary of the expected region. So that's the first part of my presentation. I hand over to Gyorgyi, and I'll be back again.
I'm back. Okay, so this was the part where we verified that the actual lab methods worked, right? So that was one set of QA/QC, if you will, and this is the part where we had to harmonize and reduce our data. So pretty much what happened was that on the left-hand side, you see that we had all these different analytical methods. And that means that for about 86 analytes, we had more than 251 source columns. So a lot of the analytes had 2 or 3 or 7 different analyses done within the database. We had to rank these analytical methods and throw out the ones that are not appropriate for the specific analyte, and then come up with a database where each analyte has exactly one value for each sample, and that value is the best that we can get from this database. So I'm not going to into a lot of details, but pretty much what we did is eliminating certain methods and ranking the remaining ones. So we threw out digestion methods that were not appropriate. So we threw out a lot of aqua regia, that are for potassium and major elements that you shouldn't really look at if you want to do lithogeochem and so on and so forth. And then we ranked the remaining methods by target level. So the results that we got, the ppm level results, are they matching with the target or with the specific method it was analyzed with? So do we have an over limit analysis for something that is not exactly over limit but overlaps with the concentration values, things like that. And then at the final run, we ranked the even remaining methods based on precision and detection limits and so on and so forth. So we came up with a database, and of course this database that we have now does have issues and limitations, and those arise because of the source data, the nature of the source data. So for example, by throwing out inappropriate digestion methods, we ended up losing data at some points. So no data is even much better than garbage data. But this reduced the numbers of samples that we can use, and inappropriate target level methods created a lot of noise. Also, this had to be done manually for each and every analyte, so this took a lot of time. And then also the merging process was very time and labor extensive. And then for the harmonized database, what we have is a lot of artifacts because of the different detection limits and precision. And so to illustrate this, I just want to show you something to understand how you should look at the data that we have now. So this is the cumulative frequency curve of a very random subsample from a much larger database that used only one digestion method, and this is for arsenic. So if you can see, like, this is a very nicely behaving curve. This is how your data should behave, and instead this is the arsenic looks like in the Malware database. And you can see that it is very choppy, it has all kinds of plateaus, and this is because we had to merge different analytical methods. Different analytical methods have different lower detection limits. So you do see that about like 40% of the data, the lowest concentration data is quite choppy, but it can be still useful. It's just we have to pay attention to these differences, right? And these artifacts. So the solution for that, we have no better choice than to indicate where we may run into problems, or we have to pay attention to this, for example, detection limits. So in the final deliverable, we have metadata that indicates whether the specific concentration data is detection limit data. We still use half of the detection limit, but some detection limits are 0.25 and some detection limits are 1 for the same element. So that may create differences in the interpretation if you're not aware of that. And also we indicate the specific analytical method that was used, so what that concentration data was acquired with. And this is for the geochemical processing. So on my part, I'm going to be doing the geochemical part, and whenever I see trends and clustering, I will have to go back and check whether this is an artificial thing or a real thing. But for the statistical processing, all these detection limit data will be imputed. So for the statistical analysis, this won't be as much of a problem. And then going to the geochemical processing, we still couldn't not start looking at the data because there's one other thing that we had to do. So we need lithogeochemical context to do the statistical analysis, right? We want to be sure that the rock we are looking at is the rock that was mapped or logged as. So first, what we had to do is dimension reduction. We have 150 something unique rock types, from typos to slight differences to whatever, so we needed to reduce that. And if you combine this with the 38 unique subclasses such as volcanic flow, mafic volcanic flow, whatever flow, so that needed a reduction. Also, doing this sort of like geochemical pre-processing is really beneficial because there are some non-lithological, it's really non-geochemical indication for rock types. For example, if you have an unknown or a schist or even a greywacke, that can be a lot of things, right? And we want to know the geochemical affinity of these rocks, so we have to bin these, like classify these. And I did something. Did I something? No? Okay. Never mind me. So, and also there are samples that are not really relevant from a lithogeochemical point of view. For example, stringer zones or veins that were analyzed for a different reason and typically have very low trace element, or at least immobile trace element concentrations, because if you have a chunky quartz vein, you won't have a lot of host rock. So those go to a different category. Those are one of the special cases. So we identified the 5 main geochemical groups that we can take and do our statistical analyses on, and further the geochemical analyses. So ultramafic, mafic, intermediate, fastic, you know, the basic things and the special cases. And also there are trends within these that I'm not going to show right now, but like there are about 24 subclasses that we can pick out, and those represent different affinities, different trace element ratios, probably different alteration trends, and so on and so forth. So this is my part. This is where I'm going to go forward with. And one of the important things to emphasize here too is that we also validated the map lithology. So there are three categories I used: confirmed, permissive, and conflict. And just to be aware of what's happening, 25% of the data is in conflict. So that means that a rock that was mapped and documented as mafic, for example, is actually a phasic rock. So this happens, but we need to know that because we are going forward with statistical analysis. And so basically this approach will be the approach that we will follow with Ahmad, who's doing most of the geoscience data, the geostatistical analysis, and on my part, who's doing the geochemical analysis, because you have to communicate, right, and go back and forth. And yeah, so now with this, we have a database that we can start working on, and I'm gonna give the mic back to Ahmed, and he's gonna start talking about the statistical part.
Okay. Well, in order to fill the gaps of this data set that we have, once the quality control finished, we have samples with below detection limit values, and we have samples for analyzed, for some analyzed. And for some analyzed, we have samples with missing values. Well, we have to estimate them or impute them to have a complete data set in order to start a statistical analysis. So, well, we have I go for the next one. I've taken 2 approaches in order to find these below detection limit values and missing values and select one of them. The first one is imputation of below value. The first approach is in 2 steps. First, we imputed the below detection limit values. The technique that I used is called ROS. Regression is abbreviation for regression on order statistics. It tries to find the distribution for the data set, and it tries to match and actually extrapolate the data below detection limit values on that distribution. Well, we ignore the missing values for the time being, and we use the technique called ROS, and we estimated or imputed actually the values below detection limit. Once it's done, we used another technique which is called Miss-Forest algorithm. This Miss-Forest algorithm estimates missing values using random forest technique and without distributional assumption. It iteratively replaces missing values with placeholders and predicts them using the model, the predicted model. So we imputed below detection limit values. We estimated missing values above detection limits. That was one procedure, one approach. The second one was using first estimating actually the missing values. We assumed that all values below detection limits are half a detection limit values, replace it with half a detection limit values. We used Miss-Forest algorithm to estimate the missing values, and then once we have the missing values, we used another algorithm and technique which is called Z composition and to estimate the below detection limit values or impute them actually. After estimating missing values, samples the below detection limit values were identified and Z composition package was used for imputation. The Z composition takes the compositional nature of the geochemical data and uses log ratio in order to estimate the values below detection limit. In order to find out which one has given better result to us, I plotted for some of elements here, cobalt, lithium, and nickel. The left graph shows the value for below detection limit estimated by Ross technique, and the right side graph shows the below detection limit values estimated by Z composition. We see that we see far better results in second approach. The Z composition has delivered much better results, continuous, and better estimation for below detection limits. So we accepted the results coming out from the Z composition, and below detection limit values imputed, above detection limit values with missing value were estimated through random forest technique. Okay, now we have a complete set of data, a full matrix, no gap. The ultimate goal of this project to us was to see if there is any difference between Wabigoon and Abitibi in chemical composition, to see if there is any, find them, and explain them, to see what the mechanism or geological phenomena has created these differences. To do that, for the data set that we have in hand, I ran K-medoid clustering. I used K-medoid to damp the effect of outliers. And after K-medoid clustering, I saw 4 clusters on that. We see that we have 4 clusters in data set. All samples are divided into 4 clusters without any labeling. If you label them to separate Abitibi samples from Wabigoon, we see that there's a difference. Well, this is a preliminary stage. Still, we are working on that. We have a definite difference in the second and third cluster. If you look at that, we see that the first cluster and the last cluster are similarity cluster. Most of the samples are divided between these 2 clusters, between Abitibi and Wabigoon. But if you look at the second and third cluster, we see, yeah, there is a difference. Most of the samples in second and third cluster are in Abitibi, and we have much less samples in this cluster sitting in Wabigoon. So it's optimistic. Yeah, we have a hope that we see some differences. Well, I have in my mind that imputation and estimation of the missing values may have created a problem, and it may have created a bias into our data set. To make sure that there is no bias on that, I did further analysis to see if there is any bias due to imputation or estimation, because we are creating the numbers based on other available numbers. Well, I used this graph separated. This shows actually the number of samples with 0 missing value versus samples with not available by clusters. For 4 clusters, we see there is no discernible pattern which emphasizes that one cluster is affected by estimation of missing values. So we are happy. For further investigation, I separated the clusters based on the number of missing value for each cluster in detail. And again, I saw no discernible pattern created due to imputation or actually estimation of the values above detection limit that we didn't have. As a conclusion, The Metal Earth's geochemical data is reliable and consistent, ensuring robust and well-supported findings. However, all interpretation must account for the quality and precision. We have to go back and check the quality control to make sure that everything is okay. If we want to do further processing, the combination of random forest and Z composition was used for imputing missing and below detection limit values. Finalizing the Metal Earth geochemistry dataset for use, no discernible pattern was observed due to the imputation process. Preliminary analysis has demonstrated the difference in the geochemical composition of Abitibi and Wabigoon. For further work, we are still doing a bit of pre-processing, rock type classification and validation. A statistical analysis of geochemical data, we have just started that. And there are more results, more interesting results, because it's just started and needs more explanation. I haven't included in this presentation. And lastly, comparison of geochemical data between Abitibi and Wabigoon, a comparative analysis between the geochemical data from these two trains will be undertaken. This study will aim to identify similarities and differences in geochemical characteristics of these regions. And that's it. Thank you.